for that matter. And if you see the diagram, it tells you you have the pluripotent stem cell or hematopoietic stem cell, and then you have the two broad cells, the myeloid progenitor, which gives rise to other innate immune cells, as you know them, and you have this lymphoid progenitor that gives rise to these T cells, the B cells, and also the natural killer cells. So it's worth of noting that actually the natural killer cells, they originate from the same progenitor cell as the T cells and the T cells. Now, in terms of maturation of the T lymphocytes, this happens more of three processes, and they can be categorized as proliferation of immature cells, and these are the pro-thymocytes that come from the bone marrow via blood. They reach the thymus region, and these are completely immature. They cannot do anything as far as immunological functions are concerned. Then we have also another stage or another process whereby these cells develop further into expression of antigen receptors. Remember, the T cells have their T cell receptors, which are very important to interact with the MHC antigen complex. So these T cell receptors also develop as part of the process of T cell maturity. Then we also have the selection of lymphocytes that express the useful antigen receptor, and that is the T cell receptor. And that process, we are going to see those processes that, or the selections that takes place in the thymus region. Now, let's look at quite a small process of thymocyte maturation. And I've said earlier on, like the prothymocytes, these are the immature T cells that leave the bone marrow via blood. They go to the thymus region and they lodge themselves there. Now, in the thymus, some things have to happen, but that is the region where these T cells matures. And these immature T cells must make their right receptors, which is very important for the immunological functionality. And that happens through the gene rearrangement. Remember, the T cell and the B cells have this T cell receptor and the B cell receptor respectively, and that gives them diversity to recognize various antigens because of the gene rearrangement that takes place alongside those receptors. Without that gene rearrangement, they cannot recognize a diverse number of antigens. And that's why, unlike the innate immune system, the adaptive immune system has this advantage of utilizing the T cell receptor as well as the B cell receptor to recognize a wide range of antigens for that particular reason. And lastly, this process, of course, like any other one, is error-prone. That means that errors do happen during the process of development, and these errors must be recognized. As much as they happen, they need to be recognized because there are implications that comes with those errors. One of the reasons is that some of these T cells might end up reacting with self-antigens and cause autoimmune diseases. And this has to be checked while it's still in the thymus region. And for that reason, there is a very elaborate process of ensuring that these cells, before they leave, they must only differentiate between self and non-self and not to cause any unnecessary immune responses, especially against the self-antigens. Now, the activities that happen in the thymus region, we can still further look at them in this way. The majority of the cells in the thymus give rise to alpha beta T cells. These are subpopulation of T cells, and these are the majority, as the statement reads. However, there are other small population of T cells. We call them gamma delta T cells, which are also part of the T cell population. So in terms of the ratio, you can say there is 95%, which are mainly due to alpha beta T cells, but there is a smaller population, which is 5%, that are composed of alpha, not alpha, but rather gamma and delta T cell receptors. Now, the development of thymocytes involves the interaction between the thymus stromal cells And these cells are actually non-hematopoietic cells. They are not blood cells, rather, but they are just tissue cells. And this, we call them the cortex and the medulla cells. And these are the two areas or critical areas that are required for the development of these T cells while in the thymus gland or the thymus region. So get to understand that the cortex is the outer part of the thymus gland, and the medulla is the inner part. And each region has a specific role that it does regarding the T cell development, and we will see what happens. Now, the earliest developing thymocytes lacked expression of the co-receptors. Remember the primary receptor, we have the T cell receptor, but within the T cell receptors, we have co-receptors, the CD4 and the CD8. The reason is that you remember that T cells are MHC restricted, meaning they only require an antigen in association with an MHC. And for that reason, when it is an antigen in association with MHC class 1, it becomes a target by the CD8 cells, or it has to be recognized by the CD8 cells. And if it's an MHC in association with class 1, then class 2, it becomes a target by the T helper cells. So the CD4 cells, as we know them, they recognize any antigen in association with class 2 molecules, while the CD8 or cytotox T cells, as we know them, they recognize an antigen that is associated with MHC class 1. So for that reason, we say that the T cells are MHC restricted, meaning they need to see an antigen in association with an MHC molecule. Now, these cells, because they lack this either of this co-receptor, whether it's CD4 or CD8, they are referred to double negative because they lack either the CD4 or the CD8 co-receptors. Okay, and we'll see at what stage this happens. Now, this double negative population can further be subdivided into various or two cells, depending on which kind of antigen they express. If they express the CD44, that is an adhesive molecule, or the CD25, which is an interleukin-2 receptor alpha gene. And these antigens are also very important, or these proteins that are on the surface, because during the maturity, even many cells, they express certain earlier antigens which disappear as the cell matures, and they develop even acquire other additional antigens in addition to what was not existing. And that's why certain things have to happen, because that is the process of development. You remember even during the tadpole, I mean the frog development, the tadpoles have the tail, but at one time when they are mature, the tail is lost. So I'm trying to give that one synonymous, like there are some antigens that develop while the cells are at the immature stage, but when the cells mature, those antigens sometimes disappear, and the cells can acquire new antigens. And that's basis of diagnosis in case of certain infections or diseases or injury, to see that there is an unexpected cells that are in the blood circulation, because they are not expected to be there anyway. Okay, because there could be a leakage or something like that. Now, the development of these CD4, CD44 cells and CD29, as we know, it gives the cells to be termed as beta selection. When they lose out these antigens, they are referred as to beta selection. And the reason is that it is part of the training, it's part of the process of development, and that shouldn't make any difference. At this stage, the cells still are developing, but it's just to recognize what is happening in terms of their outer antigens or the surface antigens. Now, the complex leads to survival, proliferation, arrest, and further alpha gene local rearrangement. And that means there's a continuous process of gene rearrangement as the cells grow, they interact with the host environment, they develop this type of interaction, and it has implication on the signal that is developed towards the development. So at one stage, these cells will acquire both co-receptors, the CD4 and the CD8. And in that way, they will be double positive. So from the double negative, the cell develop into a double positive. And from double positive, you will see later that actually these cells will develop into a single negative. So during the process of negative selection, you find that actually the T cells have to be selected or have to be allowed to go into blood circulation account that they don't react with self-antigen. So if you remember in positive, and we are going to talk about these words, positive selection, in the cortex, immediately from the thymus, from the blood, the cells migrate, they reach the cortex region. Then there they are the
Excuse me,
double
Dr.
negative. They, yes?
Excuse me. There's a comment on the chat that if you could take us a bit slowly.
Oh, okay. Okay, fine. I get the point. I'm still saying that these terminologies, we repeated them. By the end of the discussion, I'll make sure all of you at least have gotten it. This is just like an overview I'm giving, but there is a diagram that I will dwell so much on it so that this one becomes very clear. So as we have said, the T cells from the bone marrow, they are prothymocytes, meaning they are not yet mature. They are they go into the thymus region via blood, and they first interact with the cortex region. Now, what is in the cortex? The cortex provides the environment for further development so that the T cells, they develop from the double negative as they came from the bone marrow into the double positive. And now it is in the cortex region that these T cells acquire the ability to interact or recognize the self antigen. All right. In other words, they recognize an MHC molecule. So any T cell that does not recognize MHC molecule, that T cell is given the signal to die because it is not going to be of any use. That is a useless T cell. And for that reason, the T cell has to recognize an MHC molecule. The reason is this MHC molecule is going to interact with an antigen. So that same MHC molecule will recognize an antigen. So what will be of benefit if the T cell cannot recognize or see that MHC molecule? And that's why this process has to take place in the cortex. So the cell there has a double positive. It is having the co-receptor for CD4 and the CD8. And if you look at even the size of this cell, it is a little bit larger in size compared to the mature T cells. And what will happen there, once these cells have passed that test, yes, they are able to recognize the MHC molecule. Now, they should be able to, because now this is positive. Remember, the process here is positive direct, meaning they can interact strongly with the MHC molecule. And that's why they talk about the positive selection, the ability of these T cells to recognize the MHC or interact with an MHC molecule. Now, the next phase, from the cortex, the cells move to the medulla, which is towards the inner part of the thymus region. Now, the thymus has its peptides or the tissue peptides. And in addition, it has also other immune cells like the antigen presenting cells. We have the macrophages, we have the dendritic cells, and these cells have a very important role to play because remember, from the cortex region, the cells are double positive. Now, when they reach the medulla, they need to differentiate into single positive. Either a down regulation has to take place so that this cell can either mature into a CD4 cell or a CD8 cell. And for that reason, the tissue cells that are found on the medulla region, these are actually the self antigens. Some of them are MHC class 1. And those are the ones that probably give a signal that the double positive cell must now change to differentiate into a CD8 cell because NMHC class 1 will give a signal that the maturity of this cell must be a CD8. If it is interaction with MHC class 2 via the antigen presenting cells, then those cells can play a very key role in making sure that the mature cell from double positive it matures into a CD4 cell, which can be able to interact with MHC class 2. So there's a lot of processes that goes on. And that's what actually this slide is trying to demonstrate. Now, if you have not heard what I've been saying, the diagram should give you a bit of a summary of what I've just talked. Like the double negatives, these are the thymocytes that are coming from the bone marrow via the blood, which are still immature. They are reaching the thymus region. And from there, they must have different antigenic expression. And you can see the adhesive factors, the CD44 positive, and some, that is the double negative one, but it is negative for the interleukin receptor, and that is CD25. But when you look at the double negative 2, something has happened. It has retained the CD44, and now it has acquired the CD25 or interleukin receptor. Remember, the work of this interleukin is actually to activate further development of these cells. I think you remember, or you learn later, like the T cells they can produce interleukin 2 cytokine, which can still act on the same cell to activate it to perform certain functions, and we call autocrine nature of the cytokines. Okay, now in the negative 3, what you see here, now the CD44 is already lost, but the CD25 is retained. And the last stage, the double negative 4, what you see is that both the CD44 and the CD25 have been lost. And what happens, there is a combination of the pre-T cell alpha dependent with the T cell alpha chains. And for that one, that makes that cell to acquire the status of the double positive, because now they can express both the CD4 and the CD8 receptors for that reason. And when they differentiate within the modular region, you find that you have mature CD4 cells or the T helper cells and the CD8 or cytotox T cells. Now, this diagram is saying the same thing, but I'm just showing here that in the cortex region, we have the cortical epithelial cells, abbreviated as the CTE, which are very rich in self-antigens. And this one has to teach this double positive cell that it has to recognize the self-antigens. And for that one, it becomes a positive selection because it can able to interact with it is able to recognize self-antigens. The reason is, if these cells cannot recognize self-antigens, meaning they are going to cause problems later. So this is very important that it has to be checked, and that is the work of the cortical or the cortex region for that reason. Now, and in the medulla, we have said, as much as the cells have now differentiated into a mature CD4 cell or CD8 cell, there is still a problem that these cells might end up again becoming autoreactive on their own capacity as a CD4 cell or as a CD8 cell. Remember, in the previous, this cell has both co-receptors, and it is good that both can recognize these self-antigens. But here, in the medulla region, the cells have differentiated further into individual CD8 or CD4 cells. And for that reason, these cells must also be tested. Are they able to auto-react with the self-antigens? Now here, if they are highly reactive with the self-antigens, these cells are not allowed to go into blood circulation. The cells that are required in the medulla region, these are the cells that need to have low or no reaction with the self antigens. Because here they are already mature, they need to go into blood circulation. So what happens? The cells that are highly reactive with the self antigens, they are actually eliminated through the process of apoptosis. And in that way, you have very small amount of the T cells that leave the thymus region into the blood circulation, because much of the elimination takes place in the thymus region. So you can appreciate the expedited or the intricate process that happens in the thymus region, right from the positive selection coming to negative selection, and finally release of the mature T cells into the blood circulation. Now, what is the role of thymus? I've said initially that the thymus has the two lobes, which is very critical for the development of the T cells. We have the lymphocyte precursors from the bone marrow, which we call them prothymocytes. And this one subsequently are transported to the thymus via the blood to mature into the T cells. Once the T cells immigrate from the thymus and constitute the peripheral T cell repertoire, response for directing many facets of the specific immune response. And this one comes in as what are the roles of various T cell populations. We'll see each and specific role of T cells, because there are different subpopulations as we talked about. Okay. Now, there is always what we call cluster of differentiation. These are antigens that are always found on the surface of the T cells, and they are labeled in the order in which they were discovered. There is CD1, 2, 3, and so forth. I can't tell you how many are at the moment, but this is simply the proteins that differentiate them as a population of T cells. Others are being shared, others are not shared. For example, CD3, it's common molecule for for all the T cells. But the CD4 is a protein specific for T helper cells. The CD8 is a protein specific for Cytotox T-Cells. So what we are saying here is that the cluster of differentiation, abbreviated as the CD, differentiates various populations of the T cells, and that helps especially in diagnosis or in investigations. And the order in which they are labeled is in the order in which they were discovered. Okay, so we have the main arm of the T cell, which we call the T cell receptor, which is very, very important in interacting with an MHC antigen complex. And that's why we talk about the T cell MHC restriction. Okay, so what is the role of these T cell receptors? I've mentioned that these are very important in recognizing a wide variety of antigens on account that they are able to recombine. Now, when the T cell receptors recombine, they can recognize a billion or many different types of antigens because of this property of gene rearrangement. And that is synonymous with the B cell receptor specific for the B cells. Okay. Now, this T cell receptor is a heterodimer, which is non-convalently associated with the alpha, not the alpha, but the gamma, the delta, epsilon chains of the CD3 molecule. I said CD3 molecule is a shared molecule between the T cells or among the T cells. So in T cell, you will have this CD3 molecule. But beyond the CD3 molecule, you have other receptors. It's like you can have the primary receptor and you can have other secondary or the co-receptors that is unique for each type of T cell for that reason. Now, this complex nature of the T cell receptor or the CD3 molecule makes it possible to interact with an antigen MHC complex. Okay. Yeah, and if you see that diagram, it defines what we are talking about, that whenever you have an antigen presented with an MHC from an antigen presenting cell, depending on what type of class is this MHC, it can either interact with a CD4 if it's an MHC class 2, or a CD8 if it is an MHC class 1. So what you see there as CD3 is a combination of these chains. There is alpha, there is gamma, beta, epsilon chains, and these are very critical because they define what a CD3 molecule is. And that gives you the first interaction with, the second interaction with an MHC. The first one, MHC antigen complex interacts with a T cell receptor. It could be a CD4 or a CD8 receptor. And once that one interacts, then you have the CD3 molecule coming on board. And that means there is now a positive signal that will be sent. There are so many things that happen. We may not have for discussion for today, but there is a lot of activation or signaling that takes place as the diagram may depict. And that causes the T cell either to release cytokines to activate other immune cells to perform specific immune responses, either from innate immune system or from the adaptive immune system. When I say innate immune system, the T cells can secrete interferon gamma and other cytokines that can activate macrophages, or it can activate secrete cytokines that can activate B cells to produce antibodies. And if it's an allergen, it can activate the B cells to produce one specific type of immunoglobulin, and that is immunoglobulin E, that is specific for allergic responses. So the guidance on which type of response is guided by the nature of the antigen. Remember, the antigen gives you the nature of the reaction that you can expect the immune system to mount. So if, for example, it's an allergen, that becomes a target for the T helper cells type 2. If it is an infection, it becomes a target by T helper type 1. So we'll see all these types of immune cells as we move on. Now let's look at these subpopulations of T cells. Now you can appreciate that they are the main coordinators of immune response, especially when you talk about the T helper cells. But let's look at generally these T cells. They are grouped into various subpopulations depending upon their unique function that they perform. Now, the common one, I think that we are familiar, we talk about the Cytotox T-Cells, which are also known as the CD8 cells. And these are actually important for recognizing any foreign molecule when it's in association with class 1 MHC molecule. And we talk about the MHC class 1 restriction. Now, there is also the T helper cells. From the word helper, it helps other cells to perform. And that's why we talk about the T helper cells as actually the main player in terms of immune responses, because it coordinates so many immune cells in terms of activating them to differentiate into specific functional cells. Now, it only sees or recognizes an antigen presented by antigen presenting cells in 2 molecule on each surface. So that becomes the T helper cells. Now, the cytotox T cells, they are very important because they bring about the lysis and pathogens, especially the virally infected cells, or sometimes the tumor cells within our body system. You find the class 1 in all nuclei. It's your liver cell that is infected with hepatitis virus. Now, the liver cell will present with MHC class 1. And that one becomes a target by the CD8. And many viruses, as we know them, the viruses are all intracellular parasites, which must live inside the cell. So as they infect or express molecule that you find in that nucleated class. Present that particular protein or the viral proteins onto the cell surface, and that will be targeted. And sometimes the hypersensitivity or the excessive immune response due to this be the continuous lysis or due to this CD8 as opposed to the presence of the pathogenic cell. So that's why sometimes when you have chronic infection, the pathogenic cell or the CD8 cells, sometimes they also can is the tumor necrosis factor.
Excuse me, Dr. You're
can
breaking.
also depress. I'm breaking. You
Yes.
can get me now?
Yes.
Yes, I'm saying then another factor here is that the CD8 can also release this tumor necrosis factor, which can also help in depressing proteosynthesis in many target cells. Now, this MHC class 1, you find them in nucleated cells. And that's why I'm saying, remember, what are these nucleated cells? These are the somatic cells of the body. Whenever you get an infection, whether it's in your kidney, whether it's in your brain, just get to understand most of these nucleated cells, we have them and they are the majority. But few cells that are class 1, class 2, you find them in antigen presenting cells, like the monocytes or stroke macrophages, the B cells, the dendritic cells. These are antigen presenting cells. Now, the T helper cells, we have said they recognize epitopes or antigens in association with class 2 MHC. I think this is just a repeat. We also find T cells help B cells to produce antibodies, which I said earlier, depending on the nature of the antigen. Sometimes if it's an allergen, the B cells can produce immunoglobulin E. If it's a normal antigen, it can produce antibodies of different like IgM or IgG for that matter. Then let's have a look at the subsets of the T helper cells quickly. They have this function that they do. Number one, they secrete interferon gamma, which activates macrophages to become more effective at killing. And this actually happens especially in innate mechanism, whereby this interferon gamma can activate the macrophages to be more respondent to the presence of certain pathogens. In gamma, the T helper type 1 can also secrete interleukin 2, which I said earlier on. You remember the development of CD25 or interleukin 2 receptor.
Excuse me, you're still
Or
breaking
the
up.
release of this interleukin. I'm breaking up. Okay.
Yeah.
Sorry, I
Yes.
think maybe my internet is. Okay. We have the interleukin 2, which I said earlier on that it's very critical in stimulating the T cells, even though the T cells, particularly the T helper cells, can still release or secrete this interleukin 2, which can act on the same cell that is secreted. As one property of cytokines, we talk about autocrine nature. When a cell releases a cytokine, and the same cytokine acts on the same cell to activate it to produce other cytokines. Now, this T helper type 1 also produces or secretes the tumor necrosis factor, which can also stimulate the recruitment of neutrophils and monocytes to the site of infection, regarding eliminating any microbes that are foreign to the body. Then there's also the interleukin 3 that promotes expansion of immature marrow progenitors of all blood cells. And lastly, we have the granulocyte monocyte colony stimulating factor that usually acts on the progenitors in the bone marrow to increase the production of neutrophils and monocytes. So you can appreciate that as the blood development is an ongoing process, the T helper cells have a role to play, even the new generation of other immune cells, especially whenever there is infection. Like now, we talk about neutrophils. If there is a bacterial infection, what happens when you take a blood picture, you expect to see a high level of neutrophils or neutrophilia. What is the cause? There is suspicion that there is a bacterial infection, and that has caused the body to produce what? More neutrophils. But what is behind the production of these neutrophils? And that is why we are talking about the T helper cells enabling the production of more of these neutrophils by releasing this granulocyte monocyte colony stimulating factor. Now, the T helper type 1 based immune reaction helps in eliminating ingested microbes through the process of phagocytosis, and that means it activates the phagocytic cells to bring about phagocytosis as required. The interferon gamma, as we have mentioned, is the main cytokine that is usually secreted by T helper type 1. Then we have another subpopulation of T cells. You remember when we mentioned like the majority were beta, but we had like the gamma, delta, the few one, the 5%. Now the T regulatory cells falls under the few ones, the gamma delta population, and these are reserves to regulate. So they don't need to be so many as such because their role is minimal compared with the other T cells. So the T regulatory cells plays a role in regulating the CD4 and the CD8 cells so that if there is anything that is already eliminated, you don't have to have excessive immune response or exaggerated, for that matter, hypersensitivity reaction. Now, let's mention about the T helper type 2 cells. These cells, unlike the type 1, they are more of associated with allergic or hypersensitivity responses. Now, they secrete these critical cytokines or interleukins, which are very important in hypersensitivity reaction. Number one, they secrete interleukin 4, which induces the differentiation of T helper type 2 cells from naive CD4 positive precursors, and this can stimulate the B cells to produce one type of immunoglobulin, and that is immunoglobulin E, which has been greatly associated with hypersensitivity reaction, okay, or allergens. Now, this also secretes interleukin 5, which activates the mast cells. So without the interleukin 5, you may not have expedited or quick inflammatory response for that reason, because mast cells are part of the innate immune response towards injury or infection, because once these mast cells are activated, they release what we call histamine, and histamine causes vasodilation, and finally there is migration of cells from the blood from the blood to the site of infection or injury because of that release of histamine. So very critical cytokine. It also releases interleukin 6, which stimulates the synthesis of acute phase proteins, what we talk about the C-reactive proteins. And these are the earlier markers of inflammatory response. If you have had in hospitals, people, they can check when they suspect inflammatory response, they can ask for the C-reactive protein test, okay? Because this is released in the liver during the early stages of inflammatory response. Then interleukin 10 is released, and it is having a function to activate the macrophages, and sometimes it can suppress the T helper type 1 production. Other additional cytokines that can be produced by T helper type 2, we have interleukin 3, which is also produced by T helper type 1 as well, and also the granulocyte monocyte colony stimulating factor, which we have mentioned. earlier on. Now, what is common about the helper type 2 based immune reaction? We talk about this as antibody-mediated immunity because they are mostly activating the B cells to produce antibodies. And that's why we talk about the T helper type 2 is mostly based on antibody-mediated immunity or immune responses. The antibodies are needed to control extracellular pathogens. Remember, unlike the viruses, other pathogens like some bacteria, the protozoa, they may not necessarily enter inside the cell, but they can remain outside the cell as part of the infection process. So for them to be neutralized or to be killed, you need the presence of antibodies, because when you have an antibody, it will bind with that antigen, and that will attract the phagocytic cells that are antibody dependent, like the natural killer cells, the neutrophils, and the macrophages, and they will clear that infection or that antigen. So just have it in mind that the T helper type 2 is mostly targeting antibody-mediated immunity by activating B cells to produce antibodies that are required for neutralization, for opsonization, and that makes it or sometimes intoxication, neutralizing the toxins that are released by pathogens. Regulatory T cells I've mentioned, they express the CD4, the CD25, and the FOXP3 proteins. And these are the biomarkers. If you want to do an experiment with the regulatory T cells, you look for monoclonal antibodies labeled CD4 positive, CD25 positive, and for that one, you get to identify your population of regulatory cells from other populations, because these monoclonal antibodies, they are used to differentiate different populations of T cells. We have said their function is to regulate, or rather to control the effector mechanism of the other T cells. That is the CD4 cell or the CD8 cell. And these are actually necessary to maintain tolerance to self-antigens, because sometimes the cells might be reactive to its own antigens. So this signal is very important, especially to avoid hypersensitivity reaction. In summary, what have we been talking about? We talk about the two processes that are behind the T cell development or ontogenesis. And we talk about the positive and negative selection. In positive selection, this involves the selection of T cells that are functional. And we said they must recognize MHC or other self antigens while in the cortex region of the thymus. And any double positive cell that is not recognizing or that is not identifying any self antigens, these cells, they don't deserve to stay, or they don't deserve to go any further. They receive a signal to die, so they don't move beyond the cortex region. While those cells that have left the double positive, that have reached now the medulla, they have differentiated into individual CD4 positive or CD4 negative. They must face the test whether they are actually reacting with the self-antigens within the medulla region, or sometimes by interacting with the other innate immune cells that are present in the medulla region, like the dendritic cells, or the antigen-presenting cells for that matter, macrophages, and for that, and any other antigen-presenting cells. So those are the main ones that you find there. So if these cells that are a single positive are found to be autoreactive or binding strongly with the self-antigens, they are eliminated. But in the double positive stage in the cortex, the cells that are binding strongly, they are selected. But in this case, it's the reverse. So get that point very clear when you want to differentiate between positive and negative selection. So in positive selection, as we talk about it more and more, it gets more. The precursor T cells enter the thymus from the blood. They are represented with self-antigens complex with MHC molecules on the surface of cortical epithelial cells. And that's what I've said. Only those thymocytes which bind with the MHC antigen complex with adequate affinity will receive a vital survival signal. And that's what I said. They must bind strongly with self-antigens. And if they don't deserve to go to the next phase, that is in the medullary region. And here, more than 95% cells die, which means only few ones are selected from the cortex area and they move to the medullary region. Now that brings us to the negative selection of T cells. That happens in the medullary region, whereby the T cells that are autoreactive are eliminated, and those that are not autoreactive or they bind with a very low signal with the self-antigens, they're allowed to go into the blood circulation. And that is what we talk about, the positive and negative selection. So in summary, just get to understand what are these T cells, where do they develop, where do they mature, what processes takes place in double positive, what happens in double negative, and what happens in single positive, and the functions of each and every type of the T cell. I'm stopping at that point. Should you have any question or any clarification, you can ask me one question now, or if you have a question before we wind up.